Right, so monoclonal antibodies is probably the topic that confuses biology students the most. Let's look at this question here. It says, monoclonal antibodies are usually made using mouse lymphocytes. This infection produces serious symptoms in patients with poor immune system. And recently it says scientists have produced monoclonal antibodies to this infection, this disease, whatever pathogen it is, using human lymphocytes produced naturally after an infection. So what's happening here is this monoclonal antibody is attaching onto the pathogen of this disease. So we basically cloned those lymphocytes to produce a lot of these antibodies which attach onto the disease. Then it says the pathogen lives in the throat of infected patients. A sample is taken from the throat of a patient with the disease and it's transferred onto a microscope slide. And it says, how could monoclonal antibodies with a fluorescent dye be used to see these pathogens on the slide? So what you want to do is bind the fluorescent dyes to the monoclonal antibodies, and you know the antibodies are going to attach onto the pathogen because that's what they've been designed to do. So when they bind onto the pathogen and you look at them through the microscope, because it's a fluorescent dye, you'll be able to see where these pathogens are. So that's how you got to explain your answer, and it's 3 marks, so we need to say 3 points here. So first thing we can say is bind the fluorescent dye to the monoclonal antibody. Then you add it to the slide which has the pathogen on it already. Then you can say the monoclonal antibodies will bind to the pathogens and then it'll show up under the microscope.